Survive Real Life. Welcome to our genetics video. I'm Wes, this is Clark, and we'll be doing the problems from lesson 12. So I'll start with number 1. Which of the terms describes or is an example of regulatory protein? And that's repressors, the activator protein, or the cap protein, and or activators. For B, which of the terms describes or is an example of a small effector molecule, allolactose, cyclic AMP, inhibitors, or inducers? C, what are the terms described? An example of sequence found within the DNA. LAC A, LAC Z, +1 and -10. And D, which of the above is not involved in the LAC operon? We said none. I'm pretty sure they all were. E, which of the above is an example of a structural gene? LAC A and LAC Z. And there's our work. Rainbow screen. That's number 1.
Okay, number 2. Let's see. The LAC operon is regulated by 2 inducers, CAMP, allolactose. CAMP and allolactose. B, the binding site for sigma factor in RNA polymerase is called the LACP, while the cap site is the binding site for the activator that regulates the LAC operon. C, the enzyme adenyl cyclase converts ATP into CAMP. D, the -35, -10, and +1 sequences are found within the LACP portion of the LAC operon. E, the LAC-I gene encodes the LAC repressor, while the LAC-Y gene encodes lactose permease. On F, attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And G, an E. coli strain that contains 2 copies of the LAC operon is called a merozygote strain.
Number 3, suppose you're examining the LAC. Okay, I'm not going to read that. A, we put no lactose or glucose metabolism because there was no IPTG. Therefore, the repressor would always be bound. It's not a blue cell, and yeah. For B, lactose metabolism will occur. There's a lack of glucose, which means cyclic AMP will facilitate LAC operon binding at the cap site, and the cell is going to be blue. C, LAC operon is fully functioning, but the cell is not blue. D and E were both very similar. With D, we put just glucose metabolism. The LAC operon is not able to function due to lack of presence of cyclic AMP. So the cap will not bind very well to the cap sequence, and so the cell is not blue. But we do also realize that there is going to be some random binding between the cap and the cap protein, and so we're assuming that's negligible, so we didn't put that the cell would be blue, so it's not blue. For E, it's pretty much the same, but it's just like if there was a minimal amount of lactose metabolism, the cell would still not be blue because it says XGAL is absent.
For 4, we followed the video.
Pretty much.
So for A, we found that it was inducible. For B, we found that it was non-inducible. C, we found that it was constitutive. D, we found it was non-inducible. And E, we found that it was constitutive. And there's not really much work. I mean, followed the video to figure it out.
5,
which is very helpful.
Yeah, it helps 5 too, but not really. So 5A, we got there both clear. B, first one with lactose would be yellow, and then one without lactose would also be yellow, and those are both 1s. C, with lactose is 2, without is 1. D, with lactose is 1, without is 0. E, with lactose is clear, and also without lactose is clear. So those are our problems for group 12.